Hello, everyone. It's a great pleasure to be here today. I'd like to start by thanking you all for coming. My name is Shanye Yin from Albert Einstein College of Medicine. I'm an assistant professor of pathology, and I'm also the director of the pathology single cell and the bioinformatics laboratory. My lab is one of the early access sites for the G4X platform. Today, I would like to walk you through how we've been using the G4X platform in our studies, and I will show you some unpublished data. I hope this will give you a clear picture how this innovative technology can really enhance our research. Uh, Today, I will talk about our study on head and neck squamous cell carcinoma, which is actually a highly heterogeneous disease. The disease can be largely separated into two subgroups, the HPV negative group and HPV positive group. Uh, it's well established that HPV negative group are more likely to be immune cold, which means that they lack the efficient anti-tumor immune response. Accordingly, they are associated with poor prognosis. In contrast, the HPV positive cases are more likely to be immune hot. They are associated with younger patients and they usually have good prognosis. However, HPV status alone cannot predict all the high-risk patients. For example, some HPV positive cases can still have very aggressive disease. Most recently, we have performed whole genome sequencing of more than 700 HPV positive and HPV negative cases. And strikingly, we found there's a dramatic difference in the mutation spectrum associated with these two groups. And the mutated gene are affecting different signaling pathways. So this indicates some really dramatic biological differences between these two diseases. And notably, we have performed HPV capture sequencing to identify the HPV integration in the human genome. In all HPV positive cases, we found that 10% of them have HPV integration directly in PD-L1 and the PD-L2 loci, and the resulting dramatic increase of the protein expression of PD-L1 and the PD-L2. So this actually indicates a mechanism that HPV integration can create an immune suppressive environment by upregulating PD-L1 and PD-L2, adding additional complexity to the patient heterogeneity. Accordingly, we think it's really important to perform single-cell spatial analysis for a comprehensive characterization of the head and neck tumor microenvironment to address some of the most important questions in the field, including how tumor microenvironments affect prognosis and treatment response, and how does the TME change over time and upon different treatment, and how can we integrate tumor microenvironment information into the clinic practice, and can we reprogram the tumor microenvironment to enhance treatment efficacy? And this summarized the approach we use. So we have been using the G4X spatial analysis to generate single-cell spatial multiomic data for hundreds of cancers already. And my lab have developed different computational pipelines to analyze the vast amount of data. And I have separated the analysis into three different levels. So level one is single cell level. So we have developed some very efficient pipeline for cell type annotation, spatial genotyping, cell-cell distance analysis, proportion analysis, and cell-cell interaction analysis on single cell level, which I call level one. In level two, we have developed very efficient pipeline which can integrate data from tens and hundreds of patients to identify the immune niche, which are the minimal units of functional cell-cell interaction. And for the third level of analysis, we have been using this vast amount of single-cell spatial data as a huge training data set to train our different AI models. And using this model, we can directly predict gene and protein expression from H and E staining. And we have also established like a multi-module models integrating the spatial information with clinical information to identify high-risk patients. This is how it works. From every single cell spatial analysis, we can get about 1 million cells. And based on their transcription, protein and the transcription, we can do this unsupervised clustering. And based on the marker gene expression, we can quickly identify all the major cell types and put them back to the tissue section. Moreover, my lab have developed very efficient pipelines to identify subtypes of each major cell type. For example, as shown here, we have identified all the B cell subtypes based on their gene expression, including memory B cell, plasma B cell, and the germinal B cell, pre-B cell, and activated B cell, and naive B cell. I think the data speaks for itself. As you taking a closer look at the data, we see all the functional, for example, plasma B cell, they are localized around the tumor or infiltrated into the tumor. In contrast, all the pre-B and the naive B cell, they are highly enriched in this lymphoid structure. So similarly, we can do the same to identify all the T cell subgroups. We see a lot of effective memory and exhausted T cell within the tumor. This is highly consistent with the anti-tumor function. Similarly, we can identify different macrophage subtypes, and we have seen a lot of M2 macrophage, which are the anti-inflammatory and pro-tumor subtype within the tumor. We didn't see those anti-tumor M1 macrophage. This is consistent with an immunosuppressive microenvironment. We can do the same for different fibroblast subtype. And we have seen there's a unique ECM fibroblast that localized around the tumor, forming a wall, probably inhibiting the T cell and B cell from entering the tumor. We can do the subtype analysis for monocytes as well. And this is for a separate note. This is just showing you an example of all the B cell subpopulation within a lymph node with head and neck cancer metastasis. We can see all these like naive B cell that are localized in this germinal center within these lymph nodes. And all this plasma and the memory B cell, they infiltrated into the metastasis tumor and doing some anti-tumor function. And here, this is some example of the protein staining we can obtain simultaneously from the G4X platform. It can allow up to staining of 24 different proteins of very powerful technology. This is just some example. We can identify the co-staining of pan-CK tumor marker and the Ki67 of the proliferation marker. And the data look exactly like what we can get from the regular immunofluorescence assays. And here, this shows the co-localization of CD4, CD8, and the CD3, and this characterizes the co-localization of CD4 and CD8 T cells. Similarly, we can simultaneously detect CD4 and the FOXP3 and those cells with cytosol marker of CD4 and the nuclear staining of FOXP3. Those are the CD4 T regulatory T cell, which may play a very important role in anti-inflammatory and pro-tumor function within the tumor. So I think this is really a great, powerful technology. So recently we have applied this G4X spatial analysis to 20 HPV positive versus 20 HPV negative oropharyngeal cancer, which is a subtype of head and neck cancer. And as you can see here, putting them all together, and the cells are color coded by different cell types, we can identify like different cell composition within each cases. Using this data, we can directly compare different cell composition between HPV positive and negative cases. As you can see here, we see that significantly more T cell and the B cell too. This is more mature B cell within HPV positive cases versus HPV negative cases. This is consistent with the fact that HPV positive cases are more likely to be associated with immune hot microenvironment. In contrast, we see several cell types that have enhanced number in HPV negative cases, including endothelial cells, fibroblast, and myeloid cells, and the smooth muscle cells. So with this vast data, what we can analyze, how can we analyze that function? So we have been developing AI-driven computational approach, which allowed us to identify immune niche without any previous knowledge and cross tens to hundreds of patients. This is a very powerful analysis, and this is how it works from the HE staining. After we doing this G4X, we can identify all the cell types. Here we only show the major cell type. Actually, we have 50 to 60 different subtypes. And using this information across tens to hundreds of cases, we can identify the shared immune niche. So there's a limited number, for example, 12 different niche, 20 different niche across many, many different patients. And each niche will be reflecting a functional cell-cell interaction network. So apply this niche discovery to all the patient data. I think some very interesting phenotype have emerged. To me, I think the most important thing is when you look at the left, only look at the cell type. The color is very busy and it's very hard to find any specific patterns. And particularly because all the tumor cells are labeled red, then when you see the picture, most of the information are red. However, after we find immune niche, for example, different tumors may be labeled as different color based on their specific differential interaction with other immune or stroma cells. The tumors can be labeled in different color. This is very important after we discover the niche. A very unique pattern have emerged that can separate these different patients. As you can see here, I have labeled different subgroups by different color. I think just based on the color and the pattern of the color, we can separate these cases into different subgroups. I think this is very important because previously, as I mentioned, the head and neck is only roughly separated into two subgroups, HPV positive and HPV negative. And here, based on this immune niche, we can separate them into 8 different subgroups already. And based on the niche, we can easily cluster the cases based on the composition of different immune niche, in which we strongly believe they are highly relevant to the function. And using cell chart, we can identify different cells and their interactions within each niche. Again, we think this is predictive of the niche function. And from this enrichment analysis, we can tell what are the exactly subcell types enriched in each niche. And I think this is also predictive of the niche function. So we have identified like two niche that are actually highly aggressive. And just give you one example here, the subtype 1, it contains 10 different cases. But interestingly, 8 cases are actually negative, start with N. However, there are also 2 cases that are actually positive, start with a P. Although they're actually positive, their immune microenvironment is faithfully mimicking what has been observed in these highly aggressive HPV negative cases. And actually, in clinic, these two HPV positive cases are very aggressive and with both with distension metastasis. And there's no, we don't think there's any mislabeling of the sample because the HPV status have been validated by both P16 staining, which is a standard in clinic for HPV infection. And then we have also detected HPV integration into the human genome. So this indicates that HPV status alone is insufficient to expand all the high-risk patients, and we suggest the niche actually is a better marker. So this niche, the tumor niche, this purple, this is which we call niche 4. So actually this niche is actually called angiogenic and metastasis promoting niche. This is characterized by a loss of tumor, very few immune, as well as a high level of fibroblasts and blood vessel, which is further validated by this protein staining, which is generated simultaneously with the G4X platform. They have a lot of blood vessels within the tumor, which may promote the tumor proliferation and metastasis. We also have a subtype 2, which is also very aggressive. So this subtype is enriched for this bright yellow niche. And interestingly, again, this contains 5 cases and 4 cases are HPV negative, but it still contains 1 HPV positive cases containing this yellow niche. And this niche is niche 3. Actually, we call it EMT-associated niche. So this niche is characterized, again, by a lot of cancer cells, very few immune cells, and the cancer cell has a very unique gene expression profiling reflecting EMT. And this, again, can further be validated by the protein staining. So we have pan-CK marker for the tumor, this SMA, this is for blood vessel, but as well as EMT marker when it's expressing protein, in protein expression observed in tumor. And the Ki67 is highly proliferating. So merging them together, this tumor is highly EMT, is highly proliferating. We also have some other interesting niche I would like to mention. For example, niche 6, we call it stroma-rich immune exclusive niche. So this niche is characterized by a lot of tumor cell, fibroblasts, as well as a high level of T cell and B cell. However, we didn't see any interaction between tumor and these immune cells, and the protein staining exactly tells us why. As you can see here, the pan-CK, this is a tumor, and this is the SMA. It's characterized of the stroma fibroblasts around the tumor, and the CD4 show you where is the T cell. And strikingly, the T cells is largely co-localized with these fibroblasts. Looks like they are trapped within this fibroblast stroma area. Very few of them can escape and get into the tumor. So this indicates that the tumor is protected by the stroma cells, and the stroma cells is trapping the T cells, so they're excluded from the tumor. And the niche 7 is this TLS-like structure, lymphoid structure. This is characterized by high level of T cell and B cell, very few tumor, and a strong T and B cell interaction. And this is mimicking the structure of a lymphoid structure. And this is also validated by the protein staining. We have a lot of signal of CD20 for B cell and CD3 for T cell. And also there's proliferation markers for these immune cells. Also have a niche 8, which we'll call it this immune suppressive myeloid niche. So this niche is characterized by a lot of tumor cell and a strong tumor microphage interaction. And this is again validated by this immune staining generated by G4X. We have a high level of pan-CK indicating tumor, and we see also many macrophage by CD68, and there's a high proliferation marker for Ki67 for tumor. You see that a strong tumor macrophage interaction within this tumor microenvironment. This is just show you one example from a full slides spatial analysis. This is a very interesting sample. You can see from the upper right corner, there's lots of fibroblast, and from the left lower part, there are few fibroblasts. Interesting, this pattern is exactly the same with macrophage. Means macrophage and fibroblasts are co-localized in the upper right. And interestingly, both T cell and B cell are excluded from this area. And where there's no macrophage or fibroblasts, we see a high enrichment of T and B cells. So this, I think this is immediately indicative of functional regulation. For example, fibroblast and macrophage team up to erase the T cell and the B cell within the tumor microenvironment. So I think this has very important clinical implication. For example, in this 40 patient cohort, we have compared HPV positive versus HPV negative cases about the survival, and we didn't see a significant difference. This is probably because there are several HPV positive cases that are high-risk patients with poor prognosis. So we have then used the immune niche in such a way that we think the subtype 1 and 2, they are high-risk cases. So we just compare those two high-risk cases. They will also have 20 cases with other group as lower risk cases. And we already see a striking difference between the two. Even these high-risk cases contain several HPV positive cases. So this strongly indicates that HPV status alone is insufficient to identify all the high-risk patients, and looks like the immune niche is a much better idea. And finally, I'd like to talk about AI-based prediction from HPV staining. So we will be generating a large amount of this G4X spatial data. I'm talking about thousands of cases. So I think that's provide a very unique, powerful data set for AI training. So we have already established a model. We can using AI to extract features directly from HPV staining. And then we have trained them with those G4X spatial data. Then we try to directly predict protein expression or cell type using this AI model directly from HPV images. So this is example one. This just shows you even only using 40 cases as a training data set, we have already achieved very high accuracy of protein expression. You can see how similar those AI prediction with those real staining by G4X. And this in this example too, I think this is more related to translational research. We can directly highlight tumor cells from HPV staining, particularly helping to identify tumor cells within the niche, within the margin, which is otherwise very difficult to identify by human eyes. As you can see here, all the tumor cells have been labeled green, while all the other immune cells or stroma cells remain unlabeled. So as a conclusion, we think HPV status alone is insufficient to define high-risk patients, and spatial profiling of tumor niche enables more precise patient stratification, and AI model can be trained using the G4 data to directly infer gene expression from H&E images. And we strongly believe multi-module models combining information from the spatial analysis and clinic and molecular data could hold promise for predicting high-risk patients and guide precision medicine. So all the work has been performed by my talented postdoc Jianhong An and Chen Hu in the lab. And special thanks to the Singular Genomics group. We have been collaborating during the last 2 years, and I look forward for more collaboration in the future and all the funding sources. And thank you all for your attention, and I'm happy to take questions. Thank you.